Mitosis is how cells divide to make exact copies of themselves. But if the genetic instructions split unevenly, the new cell will mutate and die. Take a human skin cell preparing to divide its massive DNA library. First, it organizes that messy genetic spaghetti, packing it into dense X shapes. Each half of that X is a perfect, identical clone of the other. But these X's are just floating randomly inside the cell. How does it guarantee one clone goes left and one goes right without missing a piece? The solution? It builds a temporary web of microscopic cables from opposite ends. These cables grab the center of every X and force them to line up perfectly single file, straight down the middle. Biologists call this organized lineup metaphase. Once the alignment is perfect, these cables snap taut. They physically rip the X's down the middle, dragging one identical half to each opposite pole, acting as traction fibers in a stage called anaphase. With the clones safely separated, the cell membrane begins to pinch down the middle, wrapping each new set of DNA in its own fresh nucleus during telophase. So that original human skin cell we started with, it finally breaks completely in half, leaving you with two identical, perfectly functioning new cellular clones.